Well, thanks, Grant, for that intro. And Drew, thank you so much for being here. All of us in this room have been inspired by you and the work that you've done over many years. Of course, we didn't get a chance to learn about your work until much more recently, but I think probably or almost certainly everyone in this room has received the vaccine that has helped to save so many lives. And we're just indebted to you and so thankful for you. We're going to cover a lot of the work that you did today, but I wanted to start out by just maybe if you could tell us the moment you got the call about the Nobel Prize. Can you tell me what was the first thing you did? What was sort of going through your mind?
Yeah, so it was actually a bit of an odd event. They called Katie at 4 in the morning
Oh, wow.
and said, Katie, you and Drew won the Nobel Prize. And she said, oh, great. And then they said, do you happen to have a better number for Drew? We tried to call him and we couldn't reach him. So Katie texts me and I call her back and I said, Katie, I think they're pulling our leg. And this is some anti-vaxxer who is, you know, joking with us, and thanks for giving them my phone number. But around 5:30, which is the time you're supposed to get the call, I got a call from Stockholm. I still wasn't 100% convinced, but I waited till 6 when they did the video announcement, and then I believed it. I called my older daughter, who had just returned from her honeymoon from Sri Lanka that evening around 2 a.m.
Wow.
To DC. She hopped on a train to come up to Boston because she didn't trust that we would do things appropriately. And I called my younger daughter and she wouldn't answer her phone, and took hours before we heard from her. So the first thing I did was to tell my family.
Wow, that's awesome. Well, let's go way back in time to when you decided you wanted to become a researcher. Maybe tell us a little about what led you to want to become a researcher in the first place.
Yeah, so that was a long time ago. I don't think anybody in this room was born when I made those decisions. When I was in high school, I fell in love with math and science and went to a college where I could do science education. And during my college career, I would spend summers in research labs in Boston doing basic science research, and really fell in love with it. I had enough high school credits to graduate in 3 years, but I didn't want to. So I spent a 4th year and did a master's degree and did full-time research and wrote a thesis. And that made me want to go to medical school for an MD PhD program. So my parents would joke that nothing ever worked in the house because I always took things apart to figure out how they worked. So from a very young age, I was interested in understanding and interested in science.
Amazing. Well, we've all heard, or I think many of us have heard stories about you and Dr. Carrico and your collaboration. We've heard about a photocopier. Maybe tell us a little about this photocopier that has become so famous, and I think it's in a museum now. Tell us a little about the photocopier and how you guys first met.
Yeah, so Katie had lab space in the building next to where I moved into in late '97. And back then, the only way to read a paper was to get the journal, the physical journal, and photocopy whatever you wanted to read. They didn't have anything on the internet. So the two of us would wait for each other to finish, and we both read a lot. So we both spent many hours a day at the photocopier copying, and one of us was always there, and we would always wait for each other. And we started talking, and she did RNA, I did dendritic cells, and we started working together, and that was 7 years ago.
Wow. I'm a huge fan of in-person work. My team knows. I love being in person. This is that example. We don't use photocopiers anymore, but we do. I just love when we're all together. Maybe can you fast forward a little bit so you got a chance to spend time with Catalin Carrico, and the two of you are collaborating. Can you tell me what was the moment that was your aha moment? Was there an aha moment where it sort of all came together?
So there were lots of aha moments, and I think that's one of the reasons why we kept going. We didn't have funding, we couldn't publish papers, but we kept generating interesting data. So there were lots of bright moments. When we figured out that figured out the mechanism for why RNA was so inflammatory and how to overcome that, that was probably the first major aha moment that led to our 2005 paper. But there were lots more after that. It wasn't one step to get RNA to work. It was hundreds or thousands of steps over 25 years.
In that 2005 paper, that was the paper that was recognized with the Nobel Prize. It was that was the particular paper.
Yep.
Amazing. So now let's fast forward from 2005 to 2019. It's December of 2019, and we're all hearing about this respiratory virus. So what's going through your head when you hear about this horrible respiratory virus, that we would learn a lot about?
So I had a friend who worked at the Wuhan Institute. He had worked at Penn beforehand. And I was emailing him, once a month we would talk. And around probably September, October, he started to comment that there was a new respiratory infection that was spreading through Wuhan, and people were dying. And everybody
September,
was
October,
concerned.
is what you said?
Of 99. So well before it was announced to the world. And every time I would talk with him, it was spread further, more people were dying, his concern kept going up. So I'm in the ID division, and I would talk to the epidemiologists, and when they heard what was going on, they also got concerned and said, this sounds like a big problem. And we all had that same conclusion. So when the Chinese government released that infections were happening around December, and then released the sequence in mid-January, we immediately started making the vaccines. And so did Moderna, and so did BioNTech, and a number of other companies.
And what was that like, that race? Because of course, when you say number of companies, it was dozens, maybe even over 100 different companies.
Yeah.
What was that race like to the first vaccine?
Yeah, it wasn't really a race. So, I would talk to Ugur Sahin, who was the CEO of BioNTech, and we would share whatever reagents we had, whatever we synthesized. Nobody was racing. Everybody saw the problem and was working as fast as they could and was helping each other out to get to a solution.
Oh, I love hearing that. We love collaboration in this group. I can't remember which conversation we had, or maybe it was a talk that I heard you give, but there was something about some of your Zoom meetings and a cat. I don't know. Can you remind me what that was?
So Penn shut down, I think it was starting around March or so, for 3 months. My lab didn't shut down because we were doing coronavirus vaccine work. We just we were 25% capacity. So there was always people in my lab, but I wasn't allowed to come into work. Penn wouldn't let me come in. The people in my lab didn't want me in. It's probably a separate reason. So everything switched to Zoom. But the fact that I'm now home full time, my cat decided, he's home, I can jump on him and sleep all day. So all of my Zoom meetings had this giant fluffy white cat in my lab. And everybody got to know the cat.
And what's your cat's name?
Xander.
Xander, amazing. So Xander's the next most famous person, second
Yeah.
most famous creature,
Well,
I guess.
my mother is first after that phone call.
That's right. She became very famous. Well, I want to shift gears a little bit to the conversation that you and I had in Palm Beach. I had the honor of giving a talk at the Penn Academy where Penn's biggest donors come together, and I gave the opening session, and Drew gave the closing session that evening, and I like that order a lot more. Two other times I've had to give talks immediately following Drew. So Drew would give a talk, and then I give a talk afterwards, and the crowd goes wild when Drew finishes up, and there's a standing ovation. It's like, oh, there's still one more speaker, and I'm like, sorry guys, you have to hear about what I do with repurposing. So I much prefer to lead off the day than to close or to go after you. But it was awesome to spend time with you in Palm Beach, and we had that shuttle ride between the hotel and one of the sessions. And during that, I was telling you a bit about every cure and the repurposing work we're doing. And I was thrilled because you immediately were like, oh, absolutely, know exactly what you guys are doing with repurposing. Love that kind of work. Throughout my career, I've seen opportunities of repurposing. Maybe tell us a little bit about your thoughts on repurposing, using drugs in new ways, and just your perspectives.
I mean, you can start with the lipid nanoparticles because they're a repurposed drug. They were developed to deliver siRNAs to the liver. They're part of FDA-approved drugs. And in 2013, I guess, we asked, could we use them to deliver mRNA? Wow. I first asked for them around 2008 when they were first published, but couldn't get them. So LNPs are a repurposed drug. And Moderna caught on, BioNTech caught on, CureVac, many other companies, after we published that they worked, used them to deliver mRNA. But just the idea of screening what you've already made, what's already been in people even better, for what else they can do, makes so much more sense. The cost of developing a new drug is hundreds and hundreds of millions of dollars, from the basic science research all the way through the clinical development. Finding an FDA-approved drug, or even one that's been in good development, is so much faster and less expensive. It makes complete sense.
Well, we love hearing that, for sure, in this room. We also, in that conversation, spoke about global access, and you shared that you are so proud of the vaccine, and you're so proud that the vaccine reached so many corners of the world. But you also talked about how it didn't actually reach all corners of the world, and also that a number of other vaccines that have been developed and are in development haven't been able to reach everyone. Can you talk just a little bit about global access? It's something we also really care about.
Yeah, so I've been involved in global access since I started science. When I was at the NIH in Tony Fauci's lab, I set up a collaboration in Chiang Mai, Thailand, where we reused a building and we did research. So I trained people in Thailand and gave them a project to do, and they've done research there ever since. I've been working in Botswana for about 25 years, and we built a lab there and trained local people, and they're now doing very advanced basic science research. When the coronavirus pandemic hit, I had already been working with a group in Bangkok, Thailand for about 10 years, where I taught them how to make RNA, how to use RNA, how to design vaccines. And they had, it's called the VRC, Vaccine Research Center. They have half a dozen researchers making vaccines. When the pandemic hit, I would talk to them once a month, and the leader said, we've been talking to the government and the royalty here, and our fear is that any vaccine that's made in the United States or Europe is going to be years before it ever comes to Southeast Asia. And they said they weren't willing to wait. And they got money from the royalty, from the government, from donors to make their own vaccine. So we sat down and we designed an mRNA vaccine. This is March, so even before the vaccines had been released or tested. And then we built a GMP production site so that they had access to produce their own vaccines for clinical trials. After that, I started talking with the WHO, and I said, wouldn't it be a good idea to make GMP sites across low and middle income countries so they have access? And if you think about that, there's many different reasons to do that. One is you have GMP sites all over the place so they can make drug. But also they will stimulate, and I do a lot of work developing basic science research to use those sites. So now there are scientists in 15 different countries working on vaccines that are important to their regions, which aren't vaccines that are important to major pharma companies. They can now develop the vaccine in their labs, go to the GMP site, do clinical trials and get them approved, and then make the vaccine for their populations. Another thing that's probably even more important, I spend a lot of time in Africa. I have labs in South Africa and Botswana. But in talking with people there, they said the hesitancy and the vaccine uptake in Africa is very poor. And one of the reasons is that they don't like the idea of a lot of colonialist white people coming in and saying, take these vaccines. And having the ability to make the vaccines locally by local people, directed by local people, I think is going to increase vaccine uptake across the world.
Well, I love hearing that, and I think there's some nuggets in there for the Every Cure team. As we think about global access, it's not just about trying to educate doctors locally and engaging doctors and patients, but maybe it's about manufacturing locally. Maybe it's about really, really deep engagement. So I love that. I'll just share a quick anecdote, Drew. I don't think I've ever told you this story, but back when I was first sick just down the street of the hospital in University of Pennsylvania with Castleman disease, I was in the ICU. We couldn't figure out the diagnosis, and my dad got the phone number of a doctor at the NIH, and he didn't know who he was, but a family friend said, you got to call this doctor. Maybe he can help figure out what's wrong with your son. Maybe he can help diagnose you. And so my dad started calling this NIH doctor, and he would take my dad's calls, and they would go through my lab tests. My dad would share this lab test and that lab test, and the doctor would come up with ideas. Maybe you should do this. Maybe David has that. And these conversations went on daily for weeks and weeks. And about 6 months later, when I finally got discharged from the hospital, I had this vague memory of these conversations that I had heard my dad having, and I said, Dad, who were you talking to all those weeks? I remember you were talking to someone, and he said, I don't really know. I didn't really ask the guy too many questions. I just told him about your lab test, but I think his name's Fauci. And I said, Fauci, huh? So I went on Google, typed in Fauci. It was Tony Fauci, yeah. So your old boss and someone we all know very well. So my dad never took the time to figure out who he was, but Dr. Fauci had been taking daily calls for weeks, and he didn't know who my dad was. That's just the kind of global citizen that he is, and eventually made the diagnosis. And anyway, I've told Tony that story before, and he got a good laugh. He's like, "Yeah, I remember your dad kept calling me Fauci, but I never corrected him, and I knew he was stressed and worried about stuff." So I loved hearing you mention Dr. Fauci. I guess one question I had for you, I'm very biased. I've been at Penn since 2008. I think this is a great place and really collaborative. I've gotten a chance to see things like cellular immunotherapy from Carl June develop here. I've been able to see gene therapies blossom from the work that Jim Wilson and others have done, and then to be able to see the work that you've done with RNAs, RNA technology. Maybe can you share a little bit about, is there something about the culture here? Are there ingredients, or do we just get lucky at Penn that we happen to have 3 rock stars that were all here at the same time? I mean, just maybe share anything that you might add on that.
Yeah, I think a lot of it is the culture, and it's the collaborative nature. Uh, Probably what highlights that the best. So I did an MD PhD. What that meant is I picked a department. I went to that department and they said, here are our faculty, pick one. And I was stuck with one of the 10 microbiologists there, and I had to pick one. The way Penn does it is grad students don't go to a department. They go to a separate center, institute. They can go to literally hundreds or thousands of researchers that share interests with that institute. So there aren't limitations. Nothing is department-based. Everything is university-based. And it's not only university. You can go to NIH, you can go to any lab in the world and do your research as a Penn grad student. That's just the open-mindedness and the collaborative effort and the collaborative feeling that people share at Penn. And I think that's what makes good research. I've been working with Carl since he got here. I've been working with Jim. I've been working with Gene Bennett. I've been working with everybody who's been here a long time. It's just a collaborative place. My lab currently collaborates with close to 300 labs around the world because that's what we do. We were brought up to be collaborative, and that's what we do.
I love it. Well, this is the last question I'm going to ask, and then we'll have a little time for our colleagues. So many of us understand the potential of RNA technologies for vaccines, for example. But maybe if you could share a little bit about your thoughts on the role that RNA technologies may be able to play for rare and inherited disorders. We love rare diseases within this room. I don't know if you could share a little bit on the therapeutic side of things.
Yes, so that's a big aspect of my lab. And we've been working on it for many years. My daughter, who's 30, when she was a college student, came and wanted to do science, just to see what it was like. I couldn't put her in my lab, so I put her in Katie's lab. But I gave her a project to figure out how to target lipid nanoparticles. LNPs go to the liver, and they go to antigen-presenting cells. But if you want to deliver RNA to any other organ, cell, tissue, you have to change the LNP. And nobody had done that before. So we figured out how to target LNPs to other organs and tissues. We can target heart, brain, lungs, kidneys. We can target T cells, B cells, NK cells. Most importantly, we can target repopulating bone marrow stem cells. And why that's important, last December, 2 companies got FDA approval for a sickle cell anemia gene therapy. What they don't say is what that gene therapy does is they take out a lot of bone marrow. They culture it, they infect it with a lentivirus that delivers and corrects the gene. They give the person chemotherapy to make room, and then they give the bone marrow back. It's projected to cost $3.2 million per person. That's great in the United States. In Africa, sub-Saharan Africa, 300,000 people a year are born with sickle cell anemia. 300,000 times 3.2 million bankrupts the world. You don't have facilities to culture, to infect, to do anything across most low and middle income countries. So our idea is that if we can target repopulating stem cells, we can encode gene editing technology in the RNA. So now the cure will likely be a simple injection. So I tell people my dream, and I don't know when it's going to happen, is to grab an igloo cooler full of RNA LNPs, go anywhere in the world, line people up and give them injections to cure sickle cell anemia. Once we do that, there are thousands of other genetic diseases of the bone marrow that can be fixed. They're already doing that for liver, because that's where LNPs go. They've cured a couple of genetic diseases, or it looks like they've cured a couple of genetic diseases. And the more organs you can target, the more diseases you can cure. So it's doing two things. It's making gene therapy easy and simple, but it's also making it available to the world. Why that's important for rare genetic diseases, if you come in with a genetic disease that 10 or 100 or one person has, no company is going to be interested in making a gene therapy for that. It's hundreds of millions of dollars to do. They'll never make their money back. But if you can do it as an in vivo fast delivery where everything is standardized except the guide for the gene editing, that's simple to do.
Love it. Well, thank you so much, Drew. I want to open it up for from our colleagues to share any questions, but maybe a round of applause for Drew. What questions do you guys have?
Yeah. Hi, I'm Charlotte. I'm the research assistant for Agile here at Springfield. I just want to say, first of all, thank you so much for a very insightful talk. I definitely learned a lot. So I had a question about you. You mentioned that David asked the question, what was the race like to the COVID vaccine? And you said, well, it wasn't a race. We were actually sharing a lot of our information. And I think a big thing here is, we collaborate with a lot of different people. And my impression, and I think the impression widely across media and other areas of science whilst the vaccine was being created, was that there was this element of a race, and that maybe there was some competition. So I was wondering, what do you think kind of the disconnect was there between what you were actually doing and what you were experiencing with sharing this information, and then what we were kind of being fed as the narrative of this competition, and how maybe we can avoid that when we're collaborating with external partners?
Yeah, I think the big difference is the science and the scientists versus the media and the world. Media isn't going to get much play if they say, oh, everybody's working together.
Yeah.
They want to say, oh, well, Moderna got $1.5 billion from the government programs. All of the other companies got lots of money. They're all racing to come to a conclusion. Ultimately, companies don't share everything because they never share anything that's IP protected. But in the beginning, they shared sequences. They shared ideas for what the best immunogens would be, how to stabilize them. So I think it's just the media didn't talk to the scientists and didn't report what we were doing.
Just follow on that question. It's pretty incredible that you and your lab collaborate with 300 other groups. Sounds like a tall task. We're a collaborative organization as well, and endeavoring to work with a lot of different groups. Do you have any tips for us on how to do that effectively?
Yeah, it's difficult. As a basic scientist, I listen to everybody, and I rarely say no to a collaboration. The few times I do say no is when the idea is I think, just crazy. And even then, I'll often do it, because what do I know? I think a lot of it is just keep an open mind. And no matter how crazy an idea is, at least give it a try. How you maintain these things, a lot of it is we supply RNA LNPs to all of these labs. So that's built-in communication. It isn't simply share a few ideas and then go off and do your work. So we have a built-in system that forces us to communicate.
But also what we're trying to do is discovering new knowledge or organizing existing knowledge in a way to generate new things. And it seems like a lot of the work you've done in the lab is similarly pushing the edge of knowledge further and further. How do you think about which direction we should go in to be successful, to create meaningful knowledge, create useful knowledge for the world?
Yes, so I show a slide when I talk to usually high school students of a whack-a-mole game. And I tell them that basic research is a lot like whack-a-mole. You really have to try everything. You have to be open-minded. You have to be willing to look at everything. We never assume that we know what we're talking about and that we know the correct knowledge. You read papers, most of them, people are guessing at what the mechanisms and hypotheses are. For anything we don't understand, we don't know the mechanism, we don't know the hypothesis. And who says that the one you're thinking of is correct?
A big part of everything's value proposition is our AI platform. I would love to know, experiences we've had where we've seen AI being applied and it's really created a ton of value, or conversely, where you've seen it applied and thought, actually, this is nonsense and a red herring.
Yeah, so my lab does a lot of AI work. I think especially when you talk to lay people, you have to be careful. What I explain is AI is the ability to look at many different things. It doesn't think, it doesn't make up hypotheses, it doesn't make conclusions. It just analyzes data. So we've probably made 30,000 RNAs with different sequences. And we want to know what sequences work best with what proteins. AI can tell us that very quickly, and that's what we use it for determining optimal sequences. Probably the best AI is the AlphaFold programs that can predict protein structures better than any person can. So a lot of it is you have to use AI correctly. You don't want to write your research term papers with AI because, as people know, AI often makes things up. So I think a lot of it is just understanding what AI strengths are. I mean, as you know, it's the training that's the most important part for AI, and giving it the appropriate data and enough of it. So you have to generate that data first.
Yeah. Drew, thank you so much for spending this time and for sharing your knowledge with the world. I'm curious if you could talk about some of the people that influenced your career and your life.
Yeah, so there are so many, it's hard to include them all. I mean, I of course have to start with my parents because they encouraged me to be curious. I would take the toaster apart, and they wouldn't yell at me for breaking the toaster. They were happy that I wanted to learn. So they definitely encouraged me to be curious, to be to investigate things. I've had a lot of mentors and labs that I've been in. The person I did my PhD thesis with, Anne Marshak Rothstein, taught me a lot about doing basic science research and about immunology. Tony Fauci taught me a lot about how to deal with the world, how to deal with politics, as well as how to do research. So there have really been a lot of people that have helped along the way.
How does it feel to win a Nobel Prize? How does your life?
Yeah, it's so starting after the vaccine was released, Katie and I were getting lots of awards, and we were traveling all over the world getting awards, meeting every imaginable member of the royalty and leaders of countries. So that was kind of the prelude to the Nobel Prize. The Nobel was certainly a step above all of that. I didn't go into this to get prizes and awards or even recognition. So my family is incredibly happy because my wife can now walk into the beauty parlor when they tell her, no, we don't have any openings. And he says, well, my husband won a Nobel Prize and I'm going to a ceremony tonight and I need my hair done. But to me, I'm just back to work, doing what I've always done.
Yeah, Melanie.
Along those lines, what's the day in the life of Drew Weissman now?
I wake up early in the morning and work, and then finish late at night, try to get a little sleep, and then work more. I still travel a lot because I have collaborators all over the world, and I give talks all over the world. So last year, we were typically home 3 to 4 days a month, and the rest of the time we were traveling. I've cut that down to no more than 2 weeks a month of traveling.
So we have computer viruses, and they're all around us. But luckily, we also have very active defense systems in the digital world. Now, you mentioned that we're trying to push these treatments from several billions infections to virtually free. But that also means that it's virtually free to create all sorts of things that might not be positive treatments, but negative. How are we doing on the detection of, well, quite harmful contaminants? Do we have early warning systems? How does humanity protect against that change in technology?
Yeah, so there's lots of different levels of protection. For protection against infectious diseases, a few years ago, DARPA started a program which was a 60-day cure. And what this program was is that if you identified a new infection anywhere in the world, they wanted to be able to give us a tube of blood, have us diagnose the virus that caused it, and make monoclonal antibodies to treat the disease within 60 days. And the idea was that you have one infection in the middle of a region, you make the monoclonal antibodies and you do a ring treatment to prevent that infection from spreading. The detection of that unusual disease, we haven't gotten anywhere with. The United States, especially during the previous administration, dismantled all of its detection preparation for pandemics. And most countries have done the same because it's expensive to do that. So we're still in a state where a new infection could appear somewhere in the world, and it would likely spread pretty far before we responded to it. We've taken another approach. We're working on developing pan-vaccines. So pan-influenza, pan-coronavirus. These vaccines will work against just about all influenzas or coronaviruses that have the potential to cross over. So we might not detect the first infection, but we'll have a drug, a vaccine already made and ready to go.
Another question. You spoke briefly about, you've had a very, very long career, obviously. RNA wasn't always thought so highly. What kept you going? It takes a long time. What motivated you to say, no, this is right, we got something here?
Yeah, so my family says it's because I'm stubborn. I think what it was is that from the very beginning that Katie and I started working together, we saw the potential of RNA. It wasn't going to be a single treatment for a single disease. It had the potential to treat so many different diseases, ranging from infectious diseases to cancer, to genetic diseases, to acute and chronic diseases. The potential of it was so enormous, we just weren't willing to give up. And I think that's what kept us going.
And second question, were you surprised by the success of the RNA vaccines? Or did you kind of expect it to?
So my big worry, so in vaccines, there's standard statements that researchers make. One of them is everything works in mice. And we had developed probably 30 different pathogen vaccines that we tested in mice. 29 of 30 worked beautifully. We had complete protection. So I wasn't worried that the vaccine was going to work. My worry was whether it would work in people. And that's because many vaccines that are developed don't work in people. The classic is the DNA vaccines that work beautifully in mice. When they got to people, they were giving them 5 grams of plasmid and weren't getting protective responses. So my fear was that it worked great in mice and in every other animal we tried, but it wouldn't work in people. So when the phase 1 clinical trials came out that were toxicity trials, I saw the antibody levels there. I knew it was going to work, and I had no concern about the phase 3.
Kind of related, but yeah, I'm a data scientist. During the pandemic, I was following the numbers quite closely, and it was like everyone, it's pretty frightening, for the vaccine, the numbers, ICUs, deaths. And I remember just feeling, so relieved, excited when the vaccine rollout came out, and obviously that dropped. I was very thankful at that point. I just wonder how you felt. You started seeing the impact that was making, global.
No, exactly. I mean, I knew from the phase 1 trial that once the vaccine was out, it would work and it would protect people. But actually seeing the numbers, and the numbers were doing this for the different variants that kept coming out, and infections kept doing this, but deaths were doing this initially and then did this, and the deaths disappeared. And I would go to the ICUs before the vaccine. Literally every bed in the ICU had somebody with COVID-19, most of them on ventilators, most of them who would never get out. Now I walk into an ICU, none of them have coronaviruses.
Well, that's an amazing way, I think, to end this conversation. And a huge round of applause for doing it.